Have you ever actually stopped to think about what connects the natural world to the synthetic one? Like, what do your DNA, a potato, and a plastic PVC pipe literally have in common? I know, it sounds kind of like a trick question or maybe the setup to a bad joke. But the answer actually unlocks the absolute structural secret of life itself. And, well, modern engineering too. Because whether we're talking biology or man-made objects, all of these things rely on one surprisingly simple, universal strategy: a repeating chain structure. Welcome to today's Explainer. We're going to be diving into biological molecules, which are basically the master builders of the natural world. I want you to think of life not as some kind of incomprehensible magic, but honestly, more like a giant set of chemical Lego bricks. Because once you understand the bricks, you totally understand the entire architecture of life. So, here's our quick roadmap for the explainer. First, we'll look at the building blocks, then meet the carbohydrates, check out the shape of sugar, dive into making and breaking bonds, and finally wrap up with the sugar detective test. All right, let's jump right into section 1, the building blocks, going from elements to chains. Before we can build a complex molecule, we gotta know what our raw materials are, right? And it's actually incredible when you think about it, because the vast majority of your body, really all human bodies, are built from just 4 fundamental elements. Seriously, just 4. Hydrogen, carbon, oxygen, and nitrogen. That's the whole shebang. From just those 4 simple ingredients on the periodic table, we get the infinite mind-boggling complexity of human biology. So how do these elements actually organize themselves? Well, they start by forming what we call a monomer. Going back to our Lego analogy, a monomer is just a single individual Lego brick. It's a really simple, small molecule. On its own, yeah, it's tiny, but it has this absolutely crucial ability to join together in long chains using really strong covalent bonds. And what happens when you snap enough of those monomer bricks together? Boom, you get a polymer. The polymer is basically your completed Lego structure. It's a huge molecule, often called a macromolecule, made from tons of those small repeating monomer subunits just chained together. Stuff like polysaccharides, proteins, and nucleic acids, they're all just giant polymers built from tiny monomers. And this brings us right back to that question I asked at the start. See, nature uses polymers like cellulose in a potato, natural rubber, proteins, and nucleic acids, like your DNA. But human engineers looked at that brilliant design and basically just copied it. We use carbon-based monomers to build man-made polymers like PVC for those plastic pipes, polyester, polythene, and nylon. It's literally the exact same structural strategy, snapping small carbon-based units into long, unbreakable chains. Okay, moving on to section 2, meet the carbohydrates, our sweet subunits. Let's meet the carbohydrates. Now, as the name kind of gives away, carbohydrate, these guys are made strictly of carbon, hydrogen, and oxygen. They follow a general formula of CX H2O Y. And a super handy rule of thumb if you're trying to spot one out in the wild, their names almost always end in ose. They also dissolve really easily in water to give you a sweet-tasting solution. At their most basic monomer level, they're built from monosaccharides, which is just a fancy science way of saying one single sugar unit. Now, take a look at how beautifully organized the system is just based purely on the carbon count. If the monosaccharide has 3 carbons, we call it a triose. Got 5 carbons, that's a pentose, like the ribose and deoxyribose that literally build your genetic code. And 6 carbons, those are hexoses. That's where you find the famous ones you definitely know: glucose, fructose, and galactose. For today, these hexoses are going to be our main focus. Now, when you look at the linear structure of a glucose molecule like this, it kind of looks like a tall spine of carbon atoms. But I really want to draw your attention to those OH clusters hanging off the sides. Those are called hydroxyl groups. Keep a close eye on them, because those hydroxyl groups are essentially the sticky connection points. They're like the little plastic knobs on top of a Lego brick that actually allow these molecules to click together and interact. Which leads us perfectly into section 3, the shape of sugar, focusing on rings and isomers. So in reality, a hexose like glucose doesn't stay in a straight line. It naturally bends into a ring structure, and this is where nature gets wildly precise. Take a look at carbon number 1 over on the far right. If the hydrogen, the H, is on top, and the hydroxyl group, the OH, is on the bottom, you've got alpha glucose. But if you just swap those two around, like if the H takes the place of the OH and vice versa, it becomes beta glucose. It's 2 forms of the exact same chemical makeup, just with a different 3D arrangement. We call these isomers. And that tiny, microscopic flip completely changes how the molecule behaves in the real world. So you might be wondering, why does your body actually care about these tiny sugar rings? Well, it boils down to two massive functions. First off, they are your ultimate source of energy for respiration. I mean, the energy you're using right now to watch and process this very explainer, it comes from breaking the numerous CH bonds in these sugars, which releases a ton of energy to make ATP. And second, they're the architectural building blocks for much larger molecules. Like we mentioned earlier, the pentoses are what build your RNA and DNA. All right, let's look at how this happens in section 4, making and breaking bonds, or as I like to call it, construction and demolition. It all basically comes down to a chemical binary involving water. To build a polymer, your body uses what's called a condensation reaction. This joins two molecules together by actually removing a water molecule. But to demolish a polymer, it uses hydrolysis, which literally means splitting with water. It breaks a chemical bond by forcing a water molecule back in. So water drops out to build things up, and water forces its way in to break things down. Let's look at this in action. Here we've got two alpha glucose molecules sitting side by side. Remember those hydroxyl or OH groups we talked about earlier? Well, when these two molecules line up perfectly, their hydroxyl groups sit right next to each other. The chemistry sparks, and boom, H2O, water, drops right out of the equation. And what's left behind is this single oxygen atom acting as a bridge linking the two sugars together. That bridge is a strong covalent link called a glycosidic bond. So by joining two monosaccharides through this condensation reaction, we just built a disaccharide. In the specific case, maltose. But hey, what goes up must come down, right? This microscopic demolition derby is happening inside you every single day. When you eat a complex carbohydrate, let's say a polysaccharide like that potato we mentioned at the beginning, your body can't just absorb it directly. So right there in your stomach and intestines, hydrolysis kicks in. Your body starts adding water molecules back into those glycosidic bonds, literally smashing those long chains back down into single, easily absorbable monosaccharides. Finally, let's wrap things up with Section 5, the sugar detective test, specifically Benedict's test. Now, obviously, you can't just see a glucose molecule with the naked eye. So how do chemists in a lab actually know if a mystery solution contains these simple reducing sugars? They have to play sugar detective, and they use something called Benedict's test. It is a beautifully simple, highly visual test. First, you take your sample and heat it up with Benedict's reagent, which starts off this very clear, vivid blue color. Second, you just watch what happens. If simple sugars are in there, a chemical reaction produces a precipitate and the color will start to shift. It goes from blue to green to yellow and finally to a thick brick red or brown. That brick red color, that's your smoking gun. It is absolute proof that simple sugars are hiding in your sample. Now, there is one major biological exception that you absolutely have to remember here. Sucrose, which is just your ordinary table sugar, is a non-reducing sugar. Because of how its specific chemical bonds are all tied up, it simply won't react to Benedict's test in that same direct way. So if you just dump Benedict's reagent into a sucrose solution and heat it up, it's going to stay stubbornly, perfectly blue. So look how far we've come. We've taken the raw elements of carbon and hydrogen, built them into sugar rings, snapped them together by dropping out water, and even learned how to hunt them down in a lab. It really gives a whole new molecular meaning to that old saying, you are what you eat. Because every single carbohydrate you consume is meticulously broken down by hydrolysis into thousands of tiny glucose monomers just to be rebuilt by condensation into the very structures that power your life. So I'll leave you with this thought. Knowing that your body is this absolute masterpiece of chemical construction, which biological Lego bricks will you choose to build yourself with tomorrow? Thanks so much for joining me on this explainer, and keep exploring the amazing science of you.